For part 2 of our biochemistry project lab final assignment, we decided to present on the topic of vaccine development. In particular, we will be focusing on the science and progress behind the SARS-CoV-2 mRNA vaccine. This presentation will be brought to you by Dana and Catherine. We hope you enjoy. So first, I wanted to go over the SARS-CoV-2 virus so you can all get a better picture of what this virus is and the theory behind the mRNA vaccine that we will touch upon in later slides. So there are 4 types of viral genomes: single or double-stranded DNA, and single or double-stranded RNA. Out of these viral genomes, SARS-CoV-2 fits into the single-stranded RNA category. Specifically, it is a positive sense RNA, which we will touch upon in the next slide. And another important feature of the SARS-CoV-2 virus is, like with any other virus, the viral proteins that make up the virus. There are 4 major well-known viral proteins that make up the SARS-CoV-2 virus. Of course, there are more than 4 proteins that make up the virus, but we will just be focusing on the 4 structural proteins for now. These 4 proteins are the envelope protein, which is essential for viral maturation and production, the membrane protein, which maintains viral envelope shape, the nucleocapsid protein, which binds the RNA genome, and the spike proteins, which mediates host cell attachment. So in the previous slide, I had said SARS-CoV-2 is an RNA virus. But what is RNA and why should we focus on it? RNA is composed of a chain of nucleotides in which ribose sugars instead of deoxyribose sugars are used as building blocks for the RNA strand. RNA has nucleotides A, U, C, and G, and this sequence of nucleotides can be used to synthesize specific proteins through a process called translation. So that, in a nutshell, is what RNA is about. With the knowledge that SARS-CoV-2 depends mainly on this RNA for proliferation in a host cell, we can use mRNA vaccines to try to combat the spread of SARS-CoV-2. And it's important to quickly develop an effective mRNA vaccine because, as this image shows here, the virus is very infectious. It is an airborne virus that can be transmitted through bodily fluid droplets and direct and indirect contact. In fact, the CDC reports that SARS-CoV-2, if not having caused death by respiratory failure, can bring some long-term health problems, such as decreased lung efficiency, kidney failure, cardiac muscle inflammation, and many other issues. Therefore, in finding an mRNA vaccine for SARS-CoV-2, we can hopefully reduce death rates, decrease viral spread rates, gain some form of immunity, and maybe even resume some part of a normal lifestyle.
Now that we've reviewed the basics of RNA and the importance of developing an mRNA vaccine, we can explain how a COVID-19 mRNA vaccine actually works. So, unlike a vaccine that injects a weakened form of a virus into our cells, an mRNA vaccine provides our cells with instructions to produce proteins which will ultimately create an immune response. In this example, the mRNA gives instructions for the spike protein. First, these vaccines are injected into the muscle of our upper arm. In this image, there's a lot going on, so let's focus on the region selected. When the mRNA is released into our cells ribosomes, an organelle responsible for synthesizing polypeptides or proteins, which I've drawn as a purple oval here, reads the mRNA sequence and produces spike proteins. Once our cell has built many proteins, the mRNA is completely broken down. Although not shown here, 3 spike proteins can come together to form a spike. Spikes and protein fragments can move to the cell surface where they will be recognized by our immune system. At this point, this cell surface would resemble a SARS-CoV-2 viral cell. Moving along, let's focus on this next region of the diagram. After a cell that has produced spike proteins undergoes apoptosis, antigen-presenting cells, a kind of immune cell, not pictured here, but very important, can take up the spikes and fragments. Similarly, these fragments will move to the immune cell surface where helper T cells, another kind of immune cell, will fight against the infection. Finally, B immune cells, such as those in this image, can also bind spike proteins. If the helper T cells activate the B cells will secrete antibodies against the spike proteins. These antibodies can now bind to the SARS-CoV-2 viral spikes, prevent the virus from binding to healthy cells, and also mark the virus for destruction. If a cell does become infected, the antigen-presenting cells can activate killer T cells, yet another immune cell type, to kill any infected cells presenting spike surface proteins. As a result of receiving this kind of vaccine, our bodies can be protected from future viral infections.
There are several pros and cons to an mRNA vaccine treatment for SARS-CoV-2. So let's cover the pros first. One of the pros is that mRNA can be very rapidly produced, as all that is really needed for mRNA synthesis is knowledge of the nucleotide sequence that encodes the viral protein of interest. From there, your cells do all the work of producing the protein and mounting an immune response. This allows for large-scale vaccine production and cheapens vaccine price tags. And unlike live attenuated vaccines with weakened viruses, mRNA vaccines are completely non-living and thus thought to be safer and better for even the elderly and immunocompromised individuals. However, there are also cons to an mRNA vaccine. mRNA itself is very unstable and prone to degradation as it is single-stranded and contains ribose sugars, unlike DNA, which is more stable than RNA. Therefore, once an mRNA vaccine is made, it is best to use it right away to ensure the best results. But one way to prolong mRNA stability is by putting it in a very cold condition, made possible by low temperature freezers. However, this makes vaccine distribution more difficult and could even disadvantage low-income areas who do not have financial access to these freezers.
So a lot of the pros that were just mentioned encourage the creation of the Pfizer BioNTech vaccine, officially known as BNT162b2. According to the New York Times, this vaccine was 95% effective in test trials, launching it to the forefront of the COVID-19 vaccine race. We are including it in our presentation because it's an mRNA-based vaccine. But what's so special about it? Well, an important feature of this vaccine is its oily shell. This shell of lipid nanoparticles serves to protect the vaccine's mRNA, which can be degraded by enzymes if it's injected directly into our bodies. Once it enters our cells, this mRNA follows the same process we described earlier to create an immune response. The Pfizer BioNTech vaccine is meant to be taken in 2 doses, 21 days apart. However, it's still not clear how long the vaccine's protection lasts. Even so, as of December 11, 2020, the FDA has issued an emergency authorization for this specific vaccine. Currently, the vaccine will be available to those who are 16 years or older, and it's recommended that patients with a history of severe allergic reactions not receive the vaccine. Although it has not been approved by the FDA, this mRNA-based vaccine and the biochemistry behind it have the potential to improve the US's situation during this pandemic. Here is a quick slide with our list of references. Finally, Kathy and I just want to say thank you all for an amazing semester, and we hope everyone stays safe, happy, and healthy.